Good morning to one and all present here. I welcome you all. I welcome you all to the 8th day of the National Online Summer School on Biotechnology, organized by the Marian Star Center, St. Mary's College, Autonomous, Thoothukudi, sponsored by the Department of Biotechnology. So let us start today's program by invoking God's blessings through a prayer song. Dear participants, kindly turn on your cameras for group photo. Dear participants, kindly turn on your photo. I'm sorry, kindly turn on your cameras for group photo. Thank you, participants. Now, I am delighted to offer a hearty and warm welcome in this beautiful day. So first, I welcome the chief guest of this program, Dr. Paul David, postdoctoral researcher, Friedrich Alexander University, Erlangen, Nuremberg. Let me share a few words about him. So after completing his schooling at James School, Bihar, he did his BSc Biotechnology at Veer Kunwar Singh University, Bihar, and MSc Biotechnology at St. Joseph's College, Trichy. He worked as a senior research fellow in the Institute of Liver and Biliary Sciences, Department of Research, New Delhi, India. He completed his PhD in the University Hospital Essen, Germany. He also completed postdoctoral research in the University of Texas, USA, and in the University Hospital Erlangen, Germany, and in the topics cancer immunology and sepsis. So we are happy to have you here, sir. I also welcome our secretary, Sister Flora Mary, our principal, Dr. Sister ASJ Lucia Rose, the overall Star College coordinator, Dr. Sister Arokia Genesius Alphonse, to this program. I also extend my warm welcome to all the deans, HODs, and the students participants from various parts of India. I once again welcome you all. Thank you. Now, I hand over the session to our chief guest, Dr. Paul David. Over to you, sir.
Can you guys hear me clearly?
Yes, sir.
Yes, sir.
So I'm really thankful to the St. Mary's College and the organization which has invited me to deliver a talk on inhibitory molecules and CD8 positive T cells during. I'm also thankful Anthony Prabhu sir, like he was my lecturer during my college days in St. Joseph's Trichy, and he asked me if I could take part in this national conference happening there in Thoothukudi. So, okay, before getting into the topic, I'll talk to you guys, give you a brief introduction like what is retrovirus and why these inhibitory molecules are important for the activation of T cells, mainly CD8 T cells, which are called cytotoxic T cells, and their importance in the controlling of retroviruses. Okay, I think you all know what is HIV. It's a human immunodeficiency virus. It's a human retrovirus. And there are only two different types of human retroviruses. One is human immunodeficiency virus, which leads to AIDS, which is acute immunodeficiency syndrome. And the other one is HTLV1, and that leads to cancer and inflammatory bowel disease. So I will talk a little bit about HIV, and then I will get into an animal model, which is a really interesting model to study HIV infection in mice. And I'll try to make a story so that each one of you could follow me and understand me. And if you have any questions, you can ask me at the end. Okay. So if we talk about HIV, like I hope you all are well aware of it. Around 37 million people are infected worldwide, and there is like 1.8 million newly infected. And more than 1 million people have died because of HIV-related causes. And you all would have heard about CAR-T treatment. They're really efficient, and people are using this treatment for HIV infection. But there is no cure, and this virus persists for longer. So now the only hope to kind of cure or treat HIV infection is the development of vaccine. And so you guys have a scope now. You can develop a vaccine for HIV. Okay, so I will talk about this interesting mice model. It is called friend virus model. It's a complex of two retroviruses. One is called friend murine leukemia virus, and the other one is spleen focus forming virus. So these two are different retroviruses. So friend virus, it's a complex of two retroviruses. So what happens during friend virus is like the resistant mice, which are able to compact the virus, they develop a strong immune response during acute phase of infection. But this immune cells, they cannot completely eliminate the virus. And due to this, the virus persists for a long time, and leads to chronicity and sometimes to cancer too. Okay. So what happens during friend virus? When there is friend virus infection, the virus reaches its peak on day 7 post-infection. And then you see there is decline in the viral load, right? And this decline in the viral load is due to the activation of CD8 positive T cells. The activation of CD8 positive T cells leads to the decline in the virus. You know, although this, as I said earlier, the CD8 positive T cells, they do not eliminate the virus completely. So they do not eliminate the virus completely, and the virus persists there for longer. And similar situation happens with hepatitis B virus, hepatitis C virus, and HIV, in human. So how do these CD8 positive T cells, they eliminate the virus? They have three different mechanisms. First, by the production of antiviral cytokines like interferon gamma, TNF alpha, IL-2. So these are different cytokines which are produced by CD8 T cells and leads to the destruction or elimination of the virus. Okay. And the next is by this exocytosis pathway, where it releases some molecules like granules and perforins like granzyme B. So granzyme B and perforin, they are very important for CD8 positive T cells. So these molecules gets into the viruses and they permeabilize the virus and at the end destroys the virus. So CD8 T cells, they use these mechanisms. And the third is by fast ligand interaction. So you guys may ask, okay, CD8 T cells being like killer cells, how come the virus persists? How come the virus escapes all these things? What happens when the virus and the CD8 T cells comes in contact with each other? There's some sort of fight going on, and the fight persists longer. And at what time? And at some time, the CD8 T cells get affected, like they are functionless. And it's not like a one-step process. The fight continues, and slowly the CD8 T cells loses its functional T cell capacity, like loses the production of interferon gamma, TNF alpha, IL-2, and cytotoxic T lymphocytes and everything. And they start losing the help from CD4 cells, because CD4 cells, they are called the helper T cells, and they help the CD8 T cells. And when the CD8 T cells stops getting like a proper help from the CD4 T cells, it's get tough for CD8 T cells to fight alone, and slowly this antiviral cytokines property goes and it leads to exhaustion. But sorry, but what am I gonna talk today is like mainly about this stuff here. So when there is exhaustion, there is increase in the frequency of this molecule called PD-1. So, and this PD-1 is a very interesting molecule, like there are so many research which has labeled this molecule PD-1 as a exhaustion marker. So if there is a exhaustion of T cells or NK cells, they express a lot of this receptor PD-1 on their surface. So it's not only PD-1, there are so many different inhibitory molecules or checkpoint molecules you see. I don't know how many of you have heard about all these different molecules, but they are really interesting in designing new uh, vaccines, or there are so many clinical trials are being based on these molecules, and so many cancer clinical trials and virus clinical trials. I would say more than 100, they're based on this. And one of the main pathway is the PD-1 and its interaction to PD-L1 and PD-L2. So what has been already published, like our group in SN, when I was doing my PhD, we have shown that PD-1 is not only an exhaustion molecule, but it's a very interesting activation cell marker. So when there was friend virus infection, if you remember the cartoon from my earlier slide, when I showed that when there is friend virus infection, there is increase in the viral load. And after day 7 of post infection, there is decline in the viral load, and then there is activation of the CD8 T cells. And the activation of CD8 T cells, this leads also to the activation of PD1 positive cells, which I earlier labeled them as exhaustion marker, which is known all over the world. But our group found that most of the activated CD8 T cells, they expressed PD1, you know, and we kind of divided this cell subsets into two, like PD1 high and PD1 low. And we saw that all these PD1 highs, so you see here two different colors like black and white, and the black one is the PD1 high cells, and the white bars are the PD1 low. And these PD1 high cells, they had higher number of effector T cells. So the effector T cells are in a way the functional T cells which are effective to fight against the virus. And then we saw that this PD1 high cells, they co-expressed other co-inhibitory receptors. Okay, this LAG3, BTLA, these are other inhibitory molecules, and PD1 high cells, they expressed all these other inhibitory molecules, and they had like a very good functional property, like they had this granularity molecule called granzyme B, and this is the functional molecule. If the T cell is functional, they will have enough of granzyme B, and if they are granular, when they get into the virus and degranulate them, they should have CD107A. So these are different markers. When you get into immunology in your coming days, you will come to know about these markers one by one. So in case if you guys do not understand, please ask me in the end, okay? And then these cells like PD1 high cells, they were efficient in killing the virus. So if you see here, these are these are the positive cells which were PD1 high, and they killed the virus. And so what we did like next was like need to see for the friend virus infection, the main target of friend virus infection, like, or the reservoir where you store everything, is the erythroblast cells. So in immunology, you have markers for every different cell types. For example, for T cells, you see CD3, and for erythroblast, we look for TER119. So it's a marker for erythroblast, and this is the reservoir of friend virus infection. And we thought like, okay, the expression of PD1 was higher on CD8 T cells. What happens to the expression of its inhibitory ligand, which is PDL1, which is PD1 is program death ligand, okay? And PDL1 is the ligand for, PD1 is a receptor, sorry, and PDL1 is the ligand there. And we saw like there was no change on the expression of its ligand on the main reservoir, but we did see change of PD1 on GR1 positive cells. So GR1 positive cells are the markers for myeloid cells in the mice models. Okay, so there arose a questions like, okay, the myeloid cells population is a big population. It comprises of neutrophils, basophils, eosinophils, and then it has monocytes, dendritic cells, macrophages. Then the question was like, which myeloid cell population express this PDL1 on their cell surface? Sorry. And in order to look into that, I drew a quick cartoon to show you that from the bone marrow, you see that you will, I think you would have gone through this in your textbook. Like in the bone marrow, you have all this hematopoiesis of the blood cells where these blood cells are formed. And then there is proper progenitors for lymphoid and myeloid cells. So I'm not touching the myeloid cells, the lymphoid cells here. I'll just show you the different myeloid cells. So the myeloid cells are further divided into granulocytes, which are basophils, neutrophils, and eosinophils. And then you got monocytes, and the monocytes are further divided into macrophages and dendritic cells. And the functions of myeloid cells are phagocytosis, and they have strong immunomodulatory functions, and they present the antigens. Okay, so what am I gonna talk today? Okay, so I will mainly talk about these two ligands, three ligands, the PDL1 and PDL2. So these are the ligands for PD1. And the other one is CD270. It's the ligand for two receptors, CD160 and CD272. So the main aim of my thesis, like my PhD thesis, was to determine which myeloid cells are the target for friend virus infection, and also to analyze the expression of inhibitory ligands on cell surface of myeloid cells infected with friend virus, and to define the role of inhibitory ligands expressed on myeloid cells for the regulation of CD8 positive T cells. So in order to do all these things, so most of my work is based on flow cytometry. So this is the gating strategy, how you gate your myeloid cells in the mouse population, mouse spleen. So I use the spleen from the mouse because friend virus infection mainly targets the spleen and lymph node, as HIV targets the lymphoid organs. That's why friend virus model is a very good model to study HIV infection in mice. And to do all these things, we had a pretty fancy virus, this friend virus, and it was tagged with a green fluorescent dye called wasabi. So this wasabi was inserted in this virus, and this was done by my colleagues. So I could not explain you all this construction and the insertion methodology, like how they did it. But I was grateful and fortunate that I could receive the virus from them, which had this wasabi dye fixed to it. Okay, so here you see these gates here, different gates. So I will take you through one by one to all these different gates. So first I gated the whole leukocytes, like the whole white blood cells. And from them, I gated for the single cells. So this population here, these are all your live cells. And there's, you see here debris. So this could be your dead cells or duplets or some clogs, which are not our population of interest. Then as my main focus is on myeloid cells, I gated for CD11B, and you see here FVD. FVD is a called fixable viability dye. It's a marker for dead cells. So dead cells, they take this dye. And, and you see here 4 plus signs. So I took out T cells, which is CD3, CD19 is B cells, NK 1.1 is NK cells, and TER119 is the erythroblasts. So all these 4 different populations were not my population of interest here. So I just put everything in one fluorochrome and took them off and just gated for my myeloid. All these myeloid cells, I further gated for monocytes, then granulocytes, and the population which were negative for both, like monocytes and granulocytes, I gated for myeloid disease and macrophages. So uh, I don't know how much you guys know about flow cytometry, but it's a very interesting technique to study immunology, although there are many others. But with flow cytometry, with one sample, like with one well, you can look into, nowadays there are advanced flow cytometry. With one color, you can look, with one sample, you can look into 50 different markers, like 50 different protein expression. Okay, so after the friend virus infection, I looked into what happens to the frequency of different myeloid subpopulation. I looked into granulocytes, monocytes, macrophages, and MDCs. So here for this work of mine, I did a kinetic study. So I intravenously injected the friend virus. So I injected the virus in the tail and it went into the circulation, mainly to spleen and lymph node and bone marrow. And then after 6 days, I sacrificed the mice. On 8 day, I sacrificed the mice. I sacrificed the mice on 10 and 12 days. So I harvested the organs like bone marrow, spleen, and lymph node. But whatever I'm presenting today from the mice data, it's from spleen. So what was interesting was like after the virus infection and after day 6 post-infection, you see there is decline in the frequency of granulocytes, monocytes, macrophages, and MDCs. Then the next aim was to look into what are the frequencies of friend virus infected cells. So in a way, we can say it's like virus infected myeloid cells. So as I said in the cartoon before, like on day 7, the viral load reaches its peak, and after that there is decline. And my results also kind of confirmed, yes, on day 6, you see there is increase in the viral load, and there is increased amount of infected granulocytes, monocytes, macrophages, and MDCs on day 7. And after that, you see there is decline in the viral load. And the next aim was to look into, okay, there is infection. Our myeloid cells are infected. What happens to the expression of these 3 different ligands, which I spoke about you guys earlier? So as I mentioned before, I used this dye called wasabi, which is a green fluorescent dye, and the virus was tagged with it. So here you see 3 different colors. So the naive is like the mice which were not infected. They were left as such. And the non-infected and infected, these are from this infected mice and this granulocytes which were positive for wasabi, they are our infected cell population. And the granulocytes which were negative for wasabi, they are the non-infected cells. So you see here there is increase in, so the red dots here, you see here there is increase in the frequency of PDL1 uh, in the infected cells in comparison to the non-infected and naive cells. And you see similar pattern in monocytes, macrophages, and MDCs. And these expressions, they were significantly increased. And similar thing was observed with the another ligand of PD1, which is PDL2. And you see there is clear difference between infected and non-infected cells, as well as infected and naive cells. So you see clear difference between all these different subpopulations. And next I looked into CD270, which is an interesting molecule because not much has been known about this molecule. But with PD1 and its interaction to its 2 inhibitory ligands like PDL1 and PDL2, there are numerous clinical trials going on. And there are so many drugs in the markets which are targeting PDL1 and PDL2. But I don't remember if there are anything which is targeting this pathway. So this was pretty interesting for me. So from here onwards, I will mainly focus on this pathway, expression of CD270. So you see some sort of similar pattern of in this inhibitory ligand too, like there is increase in the infected cells on day 10 and 12, which I showed you in my previous slides. Okay, the next question was, okay, I see there is increase in the expression of CD270 on different myeloid subpopulation. What happens to the receptor? So I looked into, so as I said, receptor for PD-L1 and PDL2 is PD1, PD160 and 72. And I looked into the frequencies of this receptors on CD8 positive T cells. So these are virus-specific CD8 positive T cells. So they mainly target the virus. And as I said before, they use these three different mechanisms to kind of eliminate the virus. And you see there was increase in the expression of PD1 on day 8, 10, and 12 days post-infection. Similar pattern was seen in CD272 and CD160. Okay, so now the next aim was to focus on this interaction of CD270 to its inhibitory receptor CD160 and to CD272. And we received an interesting antibody from a group in Spain. And they had this antibody which could specifically block the interaction of CD270 to its inhibitory receptors. So after this, we planned an experiment. So we injected the mice with friend virus, and on day 6 post infection, we administered anti CD270 and anti CD160, single or in combination, and we injected it twice, and we sacrificed the mice on day post infection. And interestingly, we did not see any difference in the expansion of effector T cells or virus-specific T cells after the administration of these two different antibodies. But what was interesting that we see that there is increase in the production of granzyme B when you do a combination therapy. When you combine 72 and CD, there was not only that, we see the viral load. There was decrease in the viral load and there was increase in the production of granzyme B. So the next aim was like, how do we translate this data in human system? Okay, so we have like some PBMCs isolated from healthy volunteers. So PBMCs are peripheral blood mononuclear cells. So peripheral blood mononuclear cells, they comprise of your lymphocytes, which is mainly T cells, B cells, NK cells, and different cells from the lymphoid subpopulation. And as well as it has the monocytes, mainly monocytes. So what we did here was we stimulated the PBMCs with PHA and IL-2. These are two different stimulant, and we in vitro we did in vitro infection with HIV, and here we did a kinetic study on day 3, 5, and 7. We took the cells from the culture and then looked into different caps. So P24, you see here, this is the capsid protein on HIV infected cells. So we saw that on day 3 and day 5 and day 7, we saw like on day 7, there was increased in the percentage of infected cells. So similar to my previous mouse slides, I did a gating strategy for human myeloid cells. So I gated for the whole leukocytes, gated for single cells to take all my live cells. And then I looked into FVD, which is the marker for live cells. So these are our dead cells, which you see here. So I just gated for the live cells here. And on the live cells, I gated for monocytes, macrophages, and DCs, myeloid DCs. And in this, I looked into, like I showed in my previous slide during the mice, the percentage of infected cells. So I don't know how many of you are aware that during HIV infection, CD4 T cells are the main reservoir of HIV infection. So it made sense to look into the percentage of CD4 positive cells after HIV infection. And this is just to compare my data with other myeloid cells here. So I see like increased amount of infected monocytes, macrophages, and MDCs. So what I did before, like where I looked into the expression of different inhibitory ligands on infected myeloid cells, I did the same here. So here you see this P24. And so this P24 was conjugated with a fluorochrome. It's PE called phycoerythrin. So in future, when you work with FACS, you will come to know about all these different fluorochromes in which, with which the different antibodies or the CD markers are conjugated. So it's easier to kind of distinguish between different cell markers, which are expressed on the cell surface and intranuclear and intracellular. So here you see control and non-infected and an infected. So the control is from this thing. This is our control, and this is our infected, and this is our non-infected. So the cells which were positive for P24, these are infected. Cells which are not positive for P24, they were non-infected. So you see like the infected cells here in red, they expressed significantly higher expression of PDL1 on monocytes, macrophages, and MDCs. And similar thing was seen with CD270. I was like, okay, I saw this a really good delta of significance in the mice, but how much is it applicable in human? And when I saw that, I could see like a clear difference between infected and non-infected, as well as in the control. This was really interesting. Like we thought like, yeah, we got a new molecule which could be worked on, that there could be like, what I'm not showing here is like, there were further studies which were done, like where we combined PDL1 and the CD160 and CD272. So maybe this paper will be published soon. So if you guys are interested, you can go through that. So this, after seeing this, we thought like, oh, that's really fascinating to see like increase in the infected cells in comparison to non-infected cells. So to sum this part up, so what we learned from the friend virus was like granulocytes and monocytes are the main myeloid targets for friend virus. And we also saw that infected myeloid cells, they enhance the expression of inhibitory ligands like PD1, PDL1, PDL2, and CD272, and increase in the percentage of virus-specific T cells expressing this receptor CD160 and CD270. They were associated with the increase in the expression of its ligand CD270 on the infected cells. And as I showed you, like blocking the interaction of CD270 to its inhibitory receptor CD160 and CD272 led to enhanced production of granzyme B and decline in the viral load. And similar thing was observed with HIV infected myeloid cells. Okay, so I will take you to another interesting story, just part 2 of the story. I hope you guys remember this, like there is increase in the viral load, and then there is decrease in the viral load. There is activation of CD8 T cells. Then you see there is contraction of the CD8 T cells. But why can't the CD8 T cells completely eliminate the virus? You see there are other molecules in the system which kind of regulates things. So one is regulatory T cells. So regulatory T cells, they are the one which suppress the activation of CD8 T cells. And then there is myeloid-derived suppressor cells. So there is a lot of definitions for the myeloid-derived suppressor cells. Some say that they are immature myeloid cells. Some say that they are uh, like dysfunctional myeloid cells. So it's not yet understood properly. Okay, so I will talk about this story now. I'll try to make it short and understandable. So regulatory T cells, they have different functions, how they kind of suppress the immune cells, which are important for the body defense mechanisms. They use different inflammatory cytokines like to suppress effector T cells. Then they kind of make the T cells dysfunctional, or by playing with the granzyme and perforin, and then they do metabolic disruption by kind of depriving the cytokines, and then they target the dendritic cells by CTLA, which is another inhibitory receptor, but I'm not going to go deep in that. So, and the next interesting thing is myeloid-derived suppressor cells. So they are of two types. One is granulocytic myeloid-derived suppressor cells. The other one is monocytic myeloid-derived suppressor cells. So what we saw here, you see on day 14, there is increase in the frequencies of these myeloid-derived suppressor cells. And if you remember the figure here, on day 14, you see there is decline. After 2 weeks, there is decline in the CD8 positive T cells. So the increase in the myeloid-derived suppressor cells leads to decline in the activation of CD8 T cells. Okay, and these myeloid-derived suppressor cells, they express nitric oxide synthase, arginase, PD-L1, and CD39. So in friend virus infection, you don't see like a clear difference, but you see difference in arginase and PD-L1. So myeloid-derived suppressor cells, how do they target the T cells? They release a different reactive oxygen species, and this leads to decrease in T cell activation, CD3 gamma delta T cells, and decrease in the naive cell subset, and then it decreases the antigen presentation of disease, IL-10, and then it decreases, it suppresses the NK cells by kind of suppressing interferon gamma and its cell cytotoxicity. And you see like it suppresses like in a different way. It's not just one way which you could target and say, okay, this is how it's suppressing our CD8 T cells or CD4 cells, let's block this. But it's not. If you block one, it's going to use the other one, and that's going to be like really adverse. So you need to be really smart how you're going to block these cells. So what I'm going to tell today is like the suppression of CD8 T cells. Uh, so this is from my publication where we showed that the suppression of CD8 T cells are mainly by three different mechanisms. One is the inhibitory ligands and receptors. Then you see the Tregs, and as well as the myeloid-derived suppressor cells. So if you see here, you see PD1, PD-L1, and B6. So B6 are just the naive mice. They are not infected. So B6 are the mice without any knockouts. So in PD1 and PD-L1, so these are the mice which do not have PD1 and PD-L1. So PD1 and PD-L1 were depleted from this mice. Okay, so when you deplete or knock out PD1 and PD-L1, you see there is increase in the virus-specific T cells. Okay, and when you deplete myeloid-derived suppressor cells, you see there is increase in the virus-specific CD8 T cells. And when you deplete Tregs, okay, when you deplete Tregs, you see there is increase in the virus-specific T cells. So the next aim was like, okay, doing single depletion, we have done all these things and we have published well, okay? So the next aim was like to understand the mechanism, how is it functioning? And so we got this Treg mice. It is a very interesting mouse model to study about Tregs, regulatory T cells. In this mouse, it has enhanced GFP protein fused under the control of FOXP promoter, okay? And it has like 97% purity, like selective depletion. It can deplete up to 97%. So these mice, they do not have Tregs. So it was a very interesting model, mice to study about Tregs and CD8 T cells. So the first aim was to do a combination therapy. And here I depleted the inhibitory ligand receptor and the Tregs. And the aim was to look into what happens to the CD8 T cells and myeloid-derived suppressor cells. So after friend virus infection, the treatment started on day 7, single or in combination. And we treated the mice 3 times and before we sacrificed the mice on day 14 post-infection. So you see here, like when we, after friend virus infection, after a combination therapy, you see when you combine the therapy by depleting PD-L1, which is the inhibitory ligand, and by depleting Tregs. So DT is the diphtheria toxin, the mice which we used. You see there is increase in the frequencies of granzyme B producing CD8 positive T cells, granzyme B producing CD4 positive T cells, and then you see there is decrease in the viral load after combination therapy. So this was going really good. And then you see after combination therapy, if you kind of cut the one arm of suppression, like we have cut here T-Rex, but that did not kind of just give us a positive results, like we showed that, okay, there is decrease in the viral load, there is increase in the production of granzyme B, but we also saw there was increase in the frequencies of these two different subsets of myeloid derived suppressor cells. These were not good. And then we also saw there was increase in the expression of CD270 after combination therapy on both the different myeloid subtypes. And you know what happens when you uh, deplete in combination? There are more chances there could be an adverse effect. So you need to be really careful, like, is it really necessary to deplete in combination or in triple? So in this case, what happened after, the thing was like after 2 weeks, when we were supposed to kill the mice, we saw like the mice which underwent combination therapy, they were really sick. They were not mobile, and the mice, they didn't look good, even though their immune system was good, but that led to some adverse effect. And same was with MHC2. We saw after combination therapy, there was increase in the frequencies of MHC2 on these two different subsets, and the other ligand CD80. So this part says like when you do a combination therapy on inhibitory ligand receptors and Tregs, it leads to the enhanced production of granzyme B in both T cells and both the T cells, and improves the control of friend virus. And the combination therapy directed on PD-L1, TIM3, and Tregs, they led to the enhanced expansion of MDSCs. And this expanded MDSCs, they expressed CD270 and CD80 on their cell surface. So the next aim was to deplete inhibitory ligand and receptors and MDSCs. And to do this, we designed similar sort of experiment like the last time. We started the treatment on day 7 post-infection, treated the mice thrice before sacrificing them. We looked like, okay, when you deplete the MDSCs and inhibitory ligand and receptor, the effect was not similar like when you deplete the regulatory T cells. So the effect was more evident when you deplete inhibitory ligands and receptors. And we saw there was decline in the viral load. And most interestingly, if you remember, when we depleted in the previous set of experiment, which I showed you, when we depleted Tregs along with inhibitory ligand and receptor, which is PD-L1 and TIM3, we saw there was increase in the myeloid-derived suppressor cells. And we expected the same here. We thought, okay, when we deplete myeloid-derived suppressor cells along with the inhibitory molecules, we should see there should be increase in the FOXP3 T cells. But that was not the case. We saw this treatment was better, but not in a way, not completely, because regulatory T cells, they have an important function in maintaining the body homeostasis, like the balance of the immune system. So we saw that there was decrease in the regulatory T cells here. And to sum this part up, I can say that combination therapy directed on PD-L1, TIM3, and myeloid-derived suppressor cells, so they did not had any additional effect in comparison to PD-L1, TIM3, and Tregs, and in the production of granzyme B and control of friend virus. And the combination therapy on PD-L1, TIM3, and myeloid-derived suppressor cells, they abrogated the expansion of Tregs. So to sum everything up, so when you deplete inhibitory ligands receptors along with Tregs, you see there is increase in the granzyme B production by CD8 positive T cells. You see there is decrease in the viral load, and you see there is increase in the myeloid-derived suppressor cells. And the next set of experiments, what I showed, if you deplete inhibitory ligands and receptors along with MDSCs, you see there is increase in the CD8 positive T cells, which produce granzymes. You see decrease in the viral load, but you also see decrease in the Tregs. Okay, so that's all I have for today, and all this won't be possible if I did not had all these wonderful colleagues. So my boss during my PhD time, Ulf Dittmar. So he's the guy who's taking care of all the corona-related stuff in Western Germany. He's a smart guy. My postdoc, Gennady Zelensky, technician Tanya Werner, and the group from Spain, which gave us the antibody. So really thankful to them. And I'm thankful to my mom for helping me and bringing me up, as well as to my wife for cheering me up for every now and then. And I'm really thankful to Anthony sir for being a good mentor, and not only mentor, but also a really good friend. And thank you all for your attention. Thank you.
Dear participants, forum is open for discussion. If you have any query or questions, you can ask to the resource person. He is ready to answer your questions. Any questions? Raise your hand. The technical person will call you. Any questions? Sir, one girl posted a question, sir, in the chat box. Is this mechanism only applicable to retrovirus or all viruses?
So retroviruses are different. So if you talk about coronavirus, it's totally different. There are different viruses, viruses, RNA viruses. So this mechanism is applicable to most of the viruses. The thing is like one of the most important cells which kills the virus.
Sir, your voice is breaking, sir. Your voice is breaking.
Is it clear now?
Now clear, sir. Now clear, sir.
So I'm saying like this mechanism is applicable for most of retroviruses and the other viruses like DNA and RNA viruses, as well as for controlling the cancer. In cancers also, you see the similar mechanism of exhaustion and the T cells getting exhausted. So it's applicable for most of the things. But our main focus, as I was in the Institute of Virology while doing my PhD, so we focused on retroviruses because our group mainly worked with friend virus model. But we also saw this in melanoma patients, like the patients who have cancers, melanoma. So if you're interested, you can go through this paper called Combination Therapy of PD-L1 and Tregs. You can just put David P and PD-L1 and PD-1 and PD-L1, and you can have a look at this paper, or I can send an email of this paper to sister who contacted me.
Sir, another question is posted in the chat box, sir. Can you explain the receptors of infected cells and its ligand?
I think I did explain, right? In infected cells, they expressed PD-1, L1, and PD-L2. These are the ligands, okay? PD-L1 and PD-L2 are different ligands, and the receptor is PD-1. And then I spoke about CD270, which is an inhibitory ligand. It is also called herpes virus entry mediator, and the receptors are CD160 and CD270. Maybe I'll go to the slide one more time and show you like where I explained everything. Oh, sorry. So you see here, I spoke mainly about these two pathways. The receptor here is PD1. So almost all the receptors, they are either present on T cells, which is your CD4 or CD8 positive T cells, or they are present on NK cells. And APCs, they are antigen presenting cells, or these receptors, they can be present either on antigen presenting cells, which are your macrophages, neutrophils. They are like the professional antigen presenting cells. Then you got your B cells. They are not professional, but they are also antigen presenting cells. And most of the tumor cells, you know, like when you have a cancerous cells, they express PDL1, PDL2 on the infected tumor cells. Like when your macrophages are infected, they express all these things. When your neutrophils are infected, when your dendritic cells are infected, they express all these ligands on their cell surface.
Dr. Paul David. I'm Dr. Genesius Alphonse. So thank you so much for accepting our invitation and delivered a lecture on the particular topic to the students. Very informative and very interesting for us. Thank you so much, sir. Kindly wait 1 more minute for formal vote of thanks.
Thank you, sir. Now I call Dr. Amala Jyoti Grace to propose the vote of thanks.
Good afternoon to one and all connected here. I thank our Almighty for the abundant blessings. It gives me immense pleasure to deliver the vote of thanks for this National Online Summer School on Biotechnology organized by Marian Star Center under Star College scheme sponsored by the Department of Biotechnology, New Delhi. I immensely thank our Secretary, Reverend Sister Flora Mary, Principal Dr. A.S.J. Luciarus, and Deputy Principal Sister Teres for their continuous support and encouragement and guidance in all our efforts. I wholeheartedly thank our resource person, Dr. Paul David, postdoctoral researcher, Frederick Alexander University, Erlangen, for his valuable talk on this topic, role of inhibitory molecules in the activation of CD80 cells during retrovirus infection. Sir, your explanation is really a rich treat for us. We got a very good knowledge on the topic. Really, your session was very informative, sir. Thank you, sir. Thank you for your gracious presence and thanks for sharing your knowledge with us, sir. I owe my special gratitude to our star coordinator, Dr. Sister Genesius Alphonse, for his thoughts to organize this National Online Summer School on Biotechnology. Thank you, Sister. I extend my heartfelt thanks to our Controller of Examination, Deans, HODs, and the faculty members who are continuously supporting for carrying out this program. And also I thank all the participants for connecting us with us virtually with great enthusiasm. Thank you everyone for spending your valuable time with us. Also, I thank the silent workers who work behind the screen and taking their responsibilities on the shoulders. Thank you all. Thank you.
Thank you, David, sir.
No worries. Thank
you. Dear participants, today's quiz will be posted through Google Classroom at 6 p.m. We will meet tomorrow through Zoom platform again. Thank you. Thank you so much.
Can I leave?
Yes, sir. Yes, sir. Sure, sir. Dear participants, fill the feedback form.